I feel like kind of an imposter here talking about the mouse because my whole background's is all bacterial. And specifically, I've worked on Recombineering for a long time. But you'll see it's a very useful tool for the mouse. And what I'd like to do today is kind of give you a summary of all the tools that are out there for genome engineering, because you kind of have to know what all your tools are before you know which the best tool is. And so you don't go ahead and grab a hammer when you need a wrench. Okay, so here's my new organism. What is a transgenic? How to make a transgenic, both classical transgenesis and homologous recombination directed. What types of constructs can be made? The components that are available to put into a construct. And then how do we make the constructs? I'll spend most of my time talking about Recombineering. Gibson assembly is another option. And this is the last you'll hear about this, but this is something really to keep in mind. This is serious here. And it's something I have to remind myself. It's getting to be that a lot of these things are so cheap to get from a company that it doesn't make any sense to make it yourself. Yes, I can make it in a few days, but if I can order it from a company and it's there in 3 days, and in the meantime I can be doing some other construct, that makes a lot of sense. And so don't forget this option, because I keep forgetting it. Down here at the bottom, boosting recombination and CRISPR-Cas9. And so I don't want you to think that that's how I think about CRISPR down here off the slide. That's what your whole course is about. It's revolutionized everything. But I'll show you my worldview of how this fits into things right at the end. Okay, so I'll spend most of my time talking about these two things, but I'll cover all of these and actually a few more that aren't on there. That reminds me, if you have burning questions, please ask them. It's much easier to be a discussion, and then there's probably somebody next to you that didn't understand what I said either. So what is a transgenic? Well, there's many definitions out there, so I want to give you a few of them. So one is relating to or denoting an organism, in my case a mouse, that contains genetic material into which foreign DNA has been artificially introduced. A mouse person tends to say foreign DNA introduced into the mouse genome by microinjection into the nucleus or the cytoplasm of a fertilized ovum. The material is typically produced by genetic engineering in bacteria. So this is where I come in here. But I want to point out this term is thrown around in many ways. And you'll hear people refer to a transgenic when they mean something that's gone randomly. Okay, so it didn't go to the locus of interest, it went randomly. So that's a transgenic. And then when they use it that way, then they mean a knockout or knock-in when they're referring to a loci. So I just want you to be aware that people throw that term around and they mean very different things, and you have to be clear what they're talking about. All right, so when I was looking up definitions of transgenic, just so we're all on the same page, if you do a Google search, it's a lot of fun. So according to Google, these are transgenic organisms, and there were tons of them. I could have, well, fortunately there's some ones that are a little more relevant. These are realistic transgenics, and then these are both from the mouse. So we're more concerned with these, and these are pretty fun. Okay, how do you make a transgenic? Classical transgenesis was pronuclear injection, took a one-cell embryo, injected the DNA, we're showing it over here, and you get random integration. And what's critical here is the copy number is very variable. You take that embryo, implant in foster mothers. Once they're born, you screen many of them to see if you're getting expression of your transgene, if you're getting some kind of level that might mean something. And as you can see, it's a little bit rolling the dice. You're really not sure what you're going to get at the end of the day. This is a little more safer, homologous recombination directed. It's similar. We take embryonic stem cells, we bring in DNA, I said electroporation, there's other mechanisms to do it. Get the DNA, screen for the homologous recombinants, inject those cells into blastocyst, foster moms, screen the pups, and look for germline transmission. And this is the big thing here, is whether that happens. Okay. So let's compare these two. So this classical method, and I'll go more into what those constructs look like later. The making of it's pretty short. To make the homologous recombination, this takes longer. We'll go into all the reasons for that. Screen is long in both cases. The big difference is with this classical method, you have multiple copies. You don't know where it's going. It could cause a phenotype just by where it went. That has nothing to do with what you put in there. Expression levels can cause problems. So this, although it's longer and harder, you're more likely to have the regular levels that you're hoping to be studying and have them relevant. So just to point out, what CRISPR has done is potentially made this much shorter, this step here. And so it's made this better. Okay. What types of constructs can be made? Pretty much anything you can imagine. It could be complicated, it can take months, it can take years sometimes, but they can be made. So here's kind of a classic transgenic construct. So we have some gene of interest here. This in this case, it's a classic transgenic, so it's 5 to 20 kb at most. So this is probably a cDNA. You may want to study, which tissues it's made in, and so we put some kind of a marker gene in here, like LAC-Z, GFP, etc. What else does it contain? It contains the regulatory elements, both upstream and downstream. It has its transcription start site, translation start and stop, transcription termination. I didn't point out, it also may have an intron or two just to stabilize the RNA. So this is kind of a classic transgenic construct. And then, as I said before, injection of this leads to random integration into the host. Where it integrates and the copy number, which you cannot control, can really influence the expression of that transgene. In contrast, a back transgenic is huge, 150 to 300 kb. Again, we have your favorite gene here, but in this case, there can be enhancers tens, hundreds of kbs away from that gene that influence expression. And so you're really bringing all the material along with it. So injection of the DNA also leads to random integration, which can cause its own problems, but the copy number is much more likely to be less of an issue. And since you're bringing all the expression stuff with it, it tends to be wild type levels. Okay. So comparing these two, small, very big, they're both random. Here, if you were really lucky, you would have 5 copies. That's pretty rare. Here, you tend to be on the low end. So many fewer copies here. Regulation is normal, and I have that in quotes, only if all elements are known and included, and that's a rare case in the mouse that you can do that with such a small piece. Here, since you have the big piece, it's much more likely you're going to get good expression. Here, you're likely to have overexpression. Here, it's likely to be, tissue specific, all those things that you want. This is quite easy to assemble, and this is much more difficult, and then often they'll have a facility do it, and that's why I have a job. Okay, what kind of components go into a construct? What are the kind of tools? And I'm not covering them all. There's so many, but I'm giving you a flavor of everything out there. So your transgenic toolbox, you start with your gene, if you're doing a back, so you can have all the regulatory sequence, or a cDNA of choice. The more of your regulatory sequences you bring along, the better. Reporter alleles, there's tons of them. GFP, Cherry, LAC-Z, snap tags, clip tags, what have you, MIC tags. Another trick is transcriptional co-expression. These iris sequences, which are from viruses. And iris stands for internal ribosome entry site. Oh, this did not work. Okay, this is how my new glasses look to me right now, actually, because the prescription's wrong. So now you know how my world looks right now. So here we have two genes, and between them we have this iris sequence, and what's supposed to happen is you get transcription, ribosomes load on the first one, and with this special sequence then you get ribosomes entering here, and so you get expression of this. This is often like a GFP reporter or something, and so you're studying your favorite gene. You don't want to knock it out, but you want to know where's it expressed, for example, and then you can have your cells light up green. When you put this sequence here, wherever this is made, your second gene is made, okay? That's the idea. These are tricky. I think in the previous slide I mentioned that the first is always much more than the second. So the second you don't get much expression. Sometimes you get no expression of the second. They're a little bit complicated to work with. Here's kind of what the structure of an iris looks like. It's pretty good size, you know, anywhere from 500 to 700 base pairs. So it's a little awkward to work with, and a lot of times you put all the effort into it and it doesn't work. So that's largely been replaced by these guys. Translational co-expression, there's a bunch of them, P2A, T2A, F2A, and they all do a similar thing. It's again from a virus. It's a self-cleavage protein, and it's small. They're 19 amino acids to like 22 amino acids, so that's a much smaller cassette. And when you have these in place, I'll show again in a second here, but you leave 17 amino acids on the first protein, and you actually leave 1 amino acid on the second one. And here's a cartoon from a paper. So you have your upstream gene. I don't know why this is all, I mean, this is actually a tiny sequence. It's only like 22 amino acids. Here's the end of it. There's your downstream gene. And basically you get transcription, and then ribosomes come along. They make the first one, and then it stops. Because of this sequence, it doesn't actually join these two last amino acids. And then so this all falls up. You get your upstream protein, and then starting with this proline, you get your downstream protein. And it works very efficiently. It almost always works. It's a small cassette, and so whenever anybody comes to me now and they say they want an iris, I said, is there any reason not to use T2A? And they're like, well, what's that? It's like, well, let's use it then, because then it works. I haven't found a case where an iris, or where a T2A couldn't work in place of an iris, so we've pretty much.
Do you know if there are some patent issues with the T2? I mean, can we really use it? Or
If there's,
I work for a company.
if there's some what issues? Oh, patent issues. I don't know. I don't have to worry about that. That's an interesting question, yeah.
Okay.
If you hear anything, let me know. Because I'm curious of the answer. Not that I'm doing anything. So the other thing is inducible expression. Inducible promoters, site-specific recombination systems, which I'm not going to talk about this really, but in the context of these guys, because you can do so much with the site-specific recombination systems. So these are all from my love from the bacterial world, and there's a series of them that we use a lot. One is from the bacteriophage P1, and the protein is named CRE for cyclization recombination. It was named for what it did for the bacteriophage. And then there's this small site called LOXP that acts on, and it's just 34 bases. Similar, there's this flipase that works on the FRT site, and this is from the yeast 2-micron circle. And then a more recent one from the Francis Stewart lab is DRE, which is very similar to these others, small site, and it's, again, from another bacteriophage. These are very useful.
Sorry, Jim, what's the difference between DRE and CRE?
They're just different bacteriophages. I wouldn't say they're more efficient. There are other options, and then we often will use more than one of them at once, as I'll show you some examples. Just to give you an idea of how they work, I'm going to start over here on the right side of the slide because this is what FRT and flipase does for the yeast 2-micron circle. And so the 2-micron circle is shown here, and we have some sequences labeled here so we can see orientation. There's these 2 FRT sites, and it was found that there's this region that would flip, hence the term flipase. And it worked at this site, and you can see it was B, C, and now it's C, B. If you take, and if you follow the bouncing ball here, these guys are pointed towards each other, right? If we take them artificially and put them in line with each other, what happens is you get the same reaction, but in this case, you delete in between and then just leave the FRT site. So that's the reaction we usually use in the mouse, although this kind of thing can be used sometimes too. Okay. The important thing is all these systems work in the mouse. They work. I don't know if there's been a creature they've been tested in yet that they don't work in. It's pretty amazing, actually.
The efficiency, is it these days the same, or is flip still worse than CRE?
I think flip is still worse than CRE, yeah. But there's still, it's usable. Okay, so what way would we use these kind of things? And so I'm just going to give you one example here. We've got some big gene that we want to get rid of these 3 exons, and we only want to do it in certain tissues, and we'll get more into that in a second. And so I should have brought this up in more than one piece here. So here is the targeting construct that I spend my time building. So I would get all this sequence on. I have a couple markers here. All you need to worry about is there's something we can select against. And I have a drug resistance here. This is, say, FRT sites flanking it. And over here, I've used this to bring in these LOX-P sites flanking my region of interest. Okay, so this is the targeting construct. I can target the chromosome, selecting Neo, and get this stuff inserted. And then after a lot of southerns, et cetera, we find that it really is in there. And then we can, in this case, produce, I forgot what I said, flip, and delete Neo. So we got rid of that drug resistance. There'll be one FRT site left behind. We still have our LOX-P sites. And then now we can produce CRE when we want and get rid of these exons and making a null for that part of the gene.
Sorry, I have a question.
Yeah.
Why would you want to get rid of Neo?
You don't always have to. Sometimes there can be some expression issues. It's usually in an initron, and so normally we don't worry about it. So it really just depends. If it's showing a phenotype, we put this construct in and we start seeing something unusual, then we'll delete it just in case the drug is causing a problem. There's all kinds of signals in introns and stuff that if you're not careful, you'll find some new phenotypes. But you don't always have to get rid of it. The nice thing is you can. So again, stealing from the internet, how would we use this? So you see my genes chain here, but we got our target gene, we've got LOX-P sites around it, and in this case, there's GFP downstream. We made that mouse with a mouse that produces CRE, and then we'll talk a bit in a few minutes here about the promoter that's on CRE. We can choose what that promoter looks like, right? So it could be just a specific tissue is making CRE, and then we'll delete this out. We're showing that over here, and then produce GFP only in those tissues, for example. Okay? And then cells that don't have active CRE because it wasn't produced, they'll be unchanged. So here's an example of this gone to the extreme, and this is amazingly powerful. So this system called Brainbow, and I think there's at least 3 or 4 different versions of it right now. These are different lock sites. Some of them interact with each other, some do not. They actually have 3 copies of this in a row, so 3 RFPs, 3 yellow, 3 cyan. And it's a stochastic event, so you produce CRE for a short amount of time. You'll have some rearrangements, and it's going to be different in each cell. And according to this little key here, so if 3 of them ended up, all 3 cases end up producing this, obviously it's blue, and then you can read the list. You get all these different colors, and it's great for tracing lineages, right? So you can, in the brain, look and see which cell came from which. So that's just an example of how this kind of stuff is used. Okay, we can regulate transgene expression in time and space by regulating recombinase expression. For example, we could make a recombinase dependent knock-in. So this is back kind of the old school technology. We've got our classic transgenic construct here. And we want, so here's our target gene, and over here we have a mutant version of it. And so now we have this big construct that has our target gene flanked by LOXP sites with a stop sign in between here, and then the mutation downstream. And then we cross it with a CRE driver that, makes CRE in our tissue of choice. And when CRE is expressed, then we delete out the wild type gene, and now only the mutant is expressed. And so we can study the mutant gene only in our tissue. And this is important in cases where, this may be an essential gene for embryogenesis or something. So if you made it a mutant from the start, you would never be able to study it. So that comes in handy for things like that. Tissue-specific expression, which I've already said some. It's the same idea. Transgene is not expressed until we produce the CRE. And again, then we get expression in specific cellular subsets. Okay? Okay. Another option is inducible CRE. We have this CRE ERT2, which is CRE fused to the estrogen receptor. And the way that works is, again, so you can actually put multiple levels of control on things. Here, you're picking your promoter of choice, and maybe it's a promoter that's only made in certain tissues. And only in those certain tissues do you get expression of CRE ERT2. But even though it's those tissues, it's still off, because CRE ERT2 is in the cytoplasm and can't get to its LOXP sites until you're ready. So even though it's being expressed in your tissue of interest, not until we add tamoxifen does this come into the nucleus, and then we get recombination between those LOXP sites, and we make our mutation of interest. So we add that to the mouse, for example, just in his drinking water. So only when you're ready to look at your mutation does it happen. Okay. All right. Did I miss? Okay, I wanted to use a back as an example of the type of constructs you can do. This is going to be kind of quick and dirty without me going into the details of how you do it, and then that's what we'll get into next. So how could you put these tools together to study your favorite gene? So again, we're back to a back that we have a very large construct. We have maybe some genes or some sequences downstream and upstream that are important for expression. So we want tissue-specific expression of our gene. And so we find the back. They're all listed online. Find the one that has the right tissue-specific driver. And then it turns out in this case we looked and there's a gene over here that when it's overexpressed causes problems. And so I'm a little worried about that. Maybe this might go up to 10 copies and I really don't want to have 10 copies of that gene. So I can get rid of it on the back. Just delete that. Now I want to make the construct of my choice. And it turns out I want to have CRI under the control of my gene here. So I have a construct that I built that has CRI linked to a drug resistance. Now we can insert that. And then back to your question, we can either get rid of the drug or not. And so now we have our transgenic construct. So those are the kind of steps that we go through for building these things. Okay, so you have to put a lot of work into this construct. And in the end of the day, maybe you don't want it going randomly. So how do you avoid random integration? That's by using the homologous recombination instead of just putting it in wherever. And that's the same idea, but you just need a lot more homology. So here I usually use 4.5, anywhere 4.5 to 6 kb of homology upstream, and then 2.5 kb downstream. We often have a drug marker here. We can that if it's inserted, we can select against. And then here's the constructs we've been talking about. You could have a fusion GFP, whatever you want here. So in this case, again, we're back to single copy events at the locus of interest, okay? So making of this is more difficult, but in the end, it's much more predictable what's going to happen. And how does the targeted work? I'm not going to go through the absolute mechanism here, but just the general concept is that targeting vector I just talked about, and here's that marker I mentioned, that if this goes randomly, then that marker gets inserted, and that kills the cells. And so you can select against those. Whereas over here, on our homologies that are flanking our cassette, if it goes and finds the proper location and inserts, this part doesn't get inserted because that's non-homologous. And then so we can select, or we can enrich for those by selecting against those. There's often a gene we can select for here. In this case, it's NEO. And then what's crucial here is determining which ones are these and which one are those. And so that takes up a fair amount of time.
Sorry, Jim. The previous slide showed that you have, on the 5' you have longer homology arm than the 3'.
It really, it's one side or the other is more accurate. And we tend to have the short side near the counter-selectable marker. You can do shorter on both sides, you're just not as efficient. So it's not like a hard, fast rule that it has to be 4.5 kb on the 5' side. It could be larger on the 3' side. So, but longer is certainly better. So we've talked about all these pieces that we put together and these wonderful constructs we made, and you know the tools now, but how do you put those together? We want to have perfect regulation, we want single copy, and how do we deal with such large pieces of DNA? And I'll talk about recombineering the majority of time, and this is a technology that's been around for almost 20 years now, and I came in early on it and helped develop some of it, and it's almost being forgotten now with CRISPR. But that's unfortunate because it really is still needed and was actually pretty much as big a game changer, and you'll see why in a second here. So now I get to show my favorite organism is E. coli here. What is recombineering? Recombineering is in vivo genetic engineering, and it's in vivo in E. coli. It's recombination mediated genetic engineering, and that's where the term came from, recombineering. It's in E. coli. Actually, there's a growing list of bacteria it works in. It's catalyzed by the lambda red proteins is what I worked on. RecET, which is Francis Stewart's lab, first worked on. And then there's a whole list of similar proteins that work, including some in the herpes virus that are being worked on some. So maybe at some point we'll be able to do this in human cells. So first done by Francis Stewart and colleagues at EMBL long before I was there. All right, so the general concept is easy. This green guy here is E. coli, and I have my DNA of interest, and I have these little homologies on the end, which happen to be on my target sequence in here, and these are short, only 50 base pairs. And that's key, because 50 base pairs is very easy to order from an oligo company. So say this is a drug cassette that I want to put into this back, for example. It's a neocassette. So I order an oligo that has 50 bases of homology, and then just 20 bases of homology to to make my PCR product. Very cheap to buy 2 oligos like that. Then you're set and you have a cassette. Throw it in the cells that have been made in the correct way that make the red proteins, and then we have our recombinant. Where does this work? It works in E. coli or the other bugs I mentioned. It can work on any bacterial episome. What does that mean? Anything that will replicate in E. coli, and that includes plasmids. So some of the classic transgenic constructs I still use Recombineering on, because you have a small construct, you can still work on the plasmids. It works on backs, and that's huge. Works on viruses. So it can be used to make all these constructs we've been talking about. What are the advantages? So here's the classical way of doing it. And actually, when backs were first found, where you could make a bacterial artificial chromosome, so this contains 200 kb of mouse gene. The first problem was how do you work on it, right? You have this great mouse gene, how do you do anything with it? So here, it's actually even ignoring a step. So we have a multi-copy plasmid that has some sequence from the back already in it. So somebody had cloned this before. Now we have to find good restriction sites here to put our cassette in. So we're building some cassette. We want to clone that in. And these sites are going to be common in a back. So it's so big that restriction sites are hard to find, unique ones to do such an event. Once we have this clone sequence, then we're going to have to put this in a cell with the back and hope that this recombines onto the back. That's how they classically did it. And that sometimes works and it sometimes does not. As compared to Recombineering here, as I mentioned, we order these 2 primers. We make our cassette. We have our target back. We've skipped this step here, and we go directly into it. So in one step, basically, we have what we did on the left and may or not be able to make because of what restriction sites are available. So comparing them, Recombineering is precise to the base pair. You can modify very large molecules, not limited by restriction sites. All you need to know is the sequence. Use short homologies, around 50 base pairs, so you can order it. It turns out you can use single-stranded DNA or double-stranded DNA, and I can talk to you about that if you would like, but different topic. It's very rapid, and it can be incredibly efficient. There's cases where I can do it, and there's absolutely no selection needed. An experiment I just did recently for a researcher is they had this huge, fancy construct on a back, and it had a LOX-P site on it. And they got concerned because they were going to do something with CRE-LOX in this mouse line after they made it. And they said, oh, can you take that out? This back is 300 kb, and there's 34 bases of DNA they don't want on it. And so what I did is a Recombineering. So in a test tube, I just used an oligo, blindly deleted that, and then just diluted the cells and kept doing serial dilutions until I found a perfect one that had deleted that. And it's a frequent enough event that you could do that. I just did a couple 96 well plates and then I had their construct.
Jim, I have a question. Do you have case where you have multiple fragments on the recombination?
When you do multiple at the same time?
Yeah.
Typically, I don't do that. You could do that, but you would probably get a mess. It's usually, it's safer to do like one at a time and then confirm what you have and then do the next. But I'll get to a case here in a minute where I do multiple things at once. So it kind of depends on the details. The proteins involved. So typically, if you're to put linear DNA in E. coli, it's not very exciting. You put it in and it's gone really quick. So, but it turned out when you make these special proteins, lambda red or RecET, you do the same experiment, and then now this ended up in your target. What are these guys? So RecE was the first one worked on, so I'll put it. It's a 5' to 3' double-strand DNA. So I should have made a cartoon. So you have your double-strand DNA that we just put in. And this guy will start going like this. And actually from both ends. And then you have a single-strand annealing protein, in this case called RecT. So if you had two of these guys in the cells that have overlapping, you can see what's going to happen here. Then that RecT is very good at finding this homology here and bringing those together. So we've just joined these two molecules. It has, I told you, normally you put linear DNA in E. coli and it's immediately degraded. This system has no known exonuclease inhibitor, which is still kind of interesting. We don't quite know why. Maybe just the recombination's fast enough that the DNA is not degraded. Because the lambda red system has this little protein that specifically inhibits the exonuclease activity. So similar proteins, a 5' to 3' exo, a single-strand annealing protein. There's a whole family of these kind of proteins. And the one other thing I wanted to point out here, this guy is huge, or as we used to say in the lab, this is as big as a barn. And this is a nice little compact guy. And it turns out if you cut off most of that, then it still has the full activity for recombination. So this N-terminal, big N, we don't know for sure what all it does, but I'll show you one thing it does here in a minute. Okay, so what way can we use? Why am I talking about these guys? We can do pretty much anything you can imagine with them. So in vivo cloning, what I'm showing here is you're throwing in some sequence and a vector. This could be a plasmid. And you want to clone these genes. It's very efficient at doing that. That's the same reaction as something that's called gap repair from the yeast world. And I use gap repair all the time. This guy up here is a very large back, 300 kb of mouse DNA. And the researcher wants to study this 30 kb region or something. So I can make this little vector just by PCR and then just pull out the region of interest. And then I have this. And there's really no, you just select for this and then screen for it, and this is what you get. You can do a gene replacement. So now I've done that gap repair, and I have one gene on there I don't want to overexpress. I want to get rid of it. I could get rid of it by replacing the red gene here with the drug resistance, for example. Or I could delete a whole region, get rid of all three of those. I could make a fusion. You know, we might hook GFP to the orange gene here. And with single-strand DNA, we can very efficiently make mutants. So that example I gave you a few minutes ago of deleting a LOXP site was using an oligo. It's a very efficient event. In some cases, I can set it up and basically 75% of the colonies I pick have my mutation in it. So straight off the first plate. Another interesting reaction, again out of Francis Stewart's lab in 2012, is this will look very similar to the others. And this actually was my assay I was doing at the same time, but it shows what they found is that in vivo you can take two fragments and put them together very efficiently, and we call that in vivo fragment joining. And what's interesting is RecET is much better than red, and it turns out it's that N-terminal end. So if you do the short RecE, it looks just like red. It's not very efficient at doing this reaction. But if you take the full length one, then it's very, very good at it. And what's the reason I'm telling you about this is you can join multiple fragments at once, all in one step. So basically, this is Gibson's assembly that you do on your own, basically free. You have the cells, you induce the cells, you throw the fragments in it, it builds the plasmid for you. That's not to say we don't use Gibson assembly. If you start getting this takes 50 bases is a good homology, and if you're starting to make constructs where there's really no way to put 50 bases of homology, sometimes it's easiest just to do Gibson assembly. I think I saw in the book you guys already did Gibson assembly earlier this week. So you know this. It's basically kind of what I drew over there in a test tube. We have our homologies here. These are often 15 to 20 bases, and then we get joining. And you can do multiple fragments and get your assembly. This is something I use, even though I was born and raised on Recombineering. You just need to know all your tools, and in each case, which is the best one to use at this point. Okay, so back to CRISPR. Boosting homologous recombination, and I'll only talk about CRISPR. Put it in big letters there. I'm not talking much about CRISPR, but that doesn't mean I don't respect it. What is it doing, however? So my background is homologous recombination. I've been working on it since my PhD, and it turns out that DNA is pretty dumb. If you've got your interesting mutant here and you want to put it in the chromosome and you want it to do that, well, good luck. It almost never happens, right? 10 to the minus 6 is sometimes generous. However, in all creatures, if I damage this guy first, that goes up. And it can go up a lot. You can get to the cases where 2 to 3% of them, now we get this exchange. So a double-strand break, when this was first all being discovered, it sounds very dangerous. Why would a cell ever want to do this? Well, it doesn't want to do it. It wants to protect itself from them. Because once your DNA is broken, there can be all kinds of problems. So the cell is very good at fixing a break. Unfortunately, in like mouse cells, which is different from yeast, there's a couple different ways of dealing with a break. So now we have our break, and what tends to happen in a mouse and in many creatures is they do this non-homologous end joining. And there's a bunch of functions that are necessary for that, and then I have a question mark there because you don't know what you're going to get out. You're going to get small deletions, maybe a little insertion. That's often even true even if you have a donor DNA that you want to insert. You still get this, okay? What we want here is the homologous recombination, and there's genes that are involved in that, and we want to get this into our gene. So this donor here, this is what I've been talking on and on and on about. This is the cassette you want. You've put a lot of time and energy in it. You want it in here. We want it to go this way. So that's where CRISPR came in. It's a easily targetable double-strand break. And that sounds cheap. That's how I think about it. But it's very elegant. So as you guys know, you make Cas9 or various other systems that work also. You make your guide RNA. You can make it in vitro. You can make it in vivo. You can go in with the plasmid. So here's a single guide. A lot of people are going to dual guides now. And I won't spend any time going into this because this is what you guys are doing. But you can inject them into the cytoplasm or the pronucleus. Now you have your targeted break. And then now there's a good chance you'll get what you want. If you're doing a knockout, it's fantastic. We pretty much never fail. It's the knock-ins that are a problem. So this, if you've even seen this week, I wouldn't be surprised. So the non-homologous end joining works very well. We get our random mutants. This is impossible to see. There's a little mutant there. There's a big one there. And then if you want to do homologous things, if you want small things, and what's my definition of small? It's like less than 40 bases. So LOXP sites work, small tags, his tags. So what can be successfully inserted? That depends on the organism. Worms, it's much more efficient than in mouse. I'm talking about mouse today, so the better question is, what can be successfully inserted routinely? We try everything now, and so that's my one last big point, is that when I get a job, we try to do it the quick way, which is using CRISPR and design things, but make it in such a way that it's all set up to be going through ES cells if CRISPR fails. And so you can be trying it multiple times over here with CRISPR while you're still building it over here. If CRISPR fails, then you're down the road with your ES cells and you'll still get whatever the researcher wanted. So small tags work quite well. His tags, LOXP. I just came from a CRISPR meeting and I can't wait to try this. They're starting to put conditional knockout, like I mentioned earlier, where you have a region flanked, a floxed region with 2 LOXP sites. They're making a 2 kb single-stranded DNA, and they got 5% of the pups had LOXP both on the same allele, which is amazing. So that saves a ton of time if that works. What we're tend to do now is we're putting 1 LOXP in and then coming back and doing a second one. And even that's faster than going through the ES cells, but this would be even faster. Larger knock-ins are mixed results. The literature, as you know, is full of CRISPR papers. New papers every single day. A lot of them are bad. A lot of them are in big flashy journals, and if you look, there's one event that happened and there's a paper about it. But what you don't know is all the ones that failed, and there's so many failures. It works. I'm not trying to discourage you. It's fantastic. It's amazing. But large knock-ins are not routine yet, and nobody has solved it yet. I was just at this meeting. Still haven't. Everybody has the same problems, and everybody tends to use both hands. I'm doing CRISPR over here, and I'm doing classic methods over here to be sure something works. Inhibiting non-homologous end joining can help. Donor design can help. There's this Richardson paper. It's funny, CRISPR stuff's so fast. This is like, I think 5 months old now. And it feels like it's an old paper. It's an old result. But they just talked about which strand you use for your donor and how much homology and how much on each side. And take home I got from this meeting is this paper. They show having a certain amount of homology on one side and another amount on the other side is important. But at this meeting, some company showed that in this paper, they say, well, it has to be like that. It turns out as long as you have a longer on one side and shorter on the other, and the longer's on the 5' end, that's what matters. It doesn't matter which strand. And so that makes it a little easier, because if you tried to read this paper and design your oligos, your head will hurt for sure. But the point is there's new stuff coming out very rapidly, and the final, it's on my next slide, is you need to pay attention to the literature. Pretty much anything you can think of, you can make. It will not necessarily be easy, but you can do it. There's amazing tools out there. How best depends on the details of the construct. Sometimes having a company do part of it is the most cost efficient. And I do this. Well, you know, have a big fancy construct. It's like I'm ordering this part. And then I get that part, and then I use Recombineering to put it where I need it. So knowing all your tools and what they can do is the most important for making these decisions. And you got to keep current on the literature. It's almost like there's not enough time because there's new things coming out all the time. And these new little tricks can really save you a lot of time. And it seems like about every time I make something, I get to the end and oh, well, that would have saved a step. You know, so you need to keep current as much as you can. So thank you for your time. I just wanted to point out I'm from EMBL, and we have these various outstations. I'm in Monterritondo, and if all goes well, Barcelona will be inserted right here very soon. So thank you very much.